Thanks for letting me kick off the speaker. It's great to have this meeting in the United States, so and hopefully you'll have future meetings here too. I understand there are about 150 people coming to this meeting, so it's a great time to have a bioinformatics meeting for sure in the Bay Area. So as been alluded to, I'm obviously a medical doctor, but I'm giving a talk to a medical audience, so I always have to start with my conflicts of interest, just a few. Suffice it to say, I've started a bunch of companies. I consult for a bunch of companies. You may not want to believe another word I say over the next hour. I wouldn't blame you. But I'm most proud of the bottom right. Those are all the companies started by my students. More than half my graduate students now start companies. Even if they go into academia and in the lab, I think we really do believe that if you want to change the world, you can't just keep writing papers about it. You have to file intellectual property. And if no one licenses that discovery, you have to go and just do it. Start a company and get this to actual patients, to families. So a lot of people use this as a slide of shame. I use my slide like this as a slide of significance. Because to us, we have to do much more than just write papers about what we're doing here. So I'm very proud of this slide. All right, so we can start. So if you haven't heard, we're in the middle of a data deluge. In all fields, not just biomedicine. My favorite example of this is The Economist magazine. Here about 3 years ago, had this cover with this person collecting ones and zeros. It's raining so much ones and zeros, he's collecting the ones and zeros. And in this issue of The Economist, they announced that the human species now generates 2 zettabytes of data every year. If you forget your metric prefixes, it goes kilo, mega, giga, tera, peta, exabyte, zettabytes. And then, of course, next year will be 4 zettabytes of data because this number is doubling every 1 or 2 or 3 years. And I'll be the first to admit most of these zettabytes of data are these YouTube videos of kittens playing with pianos and yarn and, you know, cute entertainment value, but perhaps no scientific value. I don't blame you there. But there are scientific data sets in these zettabytes. So, for example, the Large Hadron Collider in Europe generated petabytes of data to help those scientists find new subatomic particles. More interesting, though, was NASA. So NASA, about a year ago, announced that they are putting online so many telescopes pointing to the sky, taking pictures, that they're predicting they may be generating an exabyte of data every day in 2020, an exabyte every day. Now, something very interesting about NASA and their exabytes right now. NASA already has so many telescopes taking pictures of the sky that there are not enough astronomers on planet Earth to look at all the pictures. So what do? So they get their friends on the internet to help. They crowdsource it. So you can go to Galaxy Zoo, you can sign up, take a test, and you can then decide, is this a star, is this a galaxy? So they have to crowdsource already to citizen scientists because there's so much scientific data, there are not enough traditional scientists to look at this data. And of course, I'm going to tell you, we are already in this field for biomedicine. We're already there. But we collect so much scientific data now that in 2007, this controversial article came out in Wired magazine, not the most scientific of journals. This is Chris Anderson, who was the editor-in-chief of Wired. And he said that we collect so much data now that the science itself is getting obsolete. Now, how do you say science is getting obsolete? Well, actually, it's a scientific method, right? So you'll remember, even in early second, third, fourth grade, fourth standard, you're taught the scientific method. Right, you come up with a question or a hypothesis, and then you go make measurements to test that hypothesis, right? That's a scientific method. But what he's saying here, where we are now, is we already have so much scientific data that the hard part is not making the measurements. The hard part, okay? You can ask people in my lab, right? 99% of my labs, the hard part is figuring out what is the question we want to ask, right? What is that question that everyone's wanted to know the answer to for, I don't know, 100 years. And no one realized you can ask that question right now because the data's already there. So the hard part now is figuring out the question. And this is controversial, especially when I give talks like this. I don't think the hard part is cloud computing. I don't think the hard part is writing code. I don't think the hard part is algorithms. The hard part in biomedical informatics is figuring out what's the biomedical question that we want to ask. And it's not this kind of field anymore where we can build tools and wait for someone to ask their question. Nobody out there gets this. It comes down to us to do this, our community. We cannot just keep building tools and hope someone knows how to ask the right questions. It's going to come down to you, and audiences like this, to ask the questions. So people call this big data in biomedicine. I'll use big data at the beginning, at the end, but otherwise, obviously, it's a buzzword. You know, it's the next big scientific revolution, but you know, this whole idea of data-driven science. Nature says that it's shameful if we don't use data. Science says we need to make data maximally available. And even the Lancet says that if you don't share research data, it's going to be an impediment to public health. I mean, this is how serious we're taking big data. Now, obviously, big cover articles now in Nature and Science and all the rest on big data. Where does all this data come from? It comes from devices like this. Now, I'm just showing you my favorite of these devices. This is an Affymetrix gene chip on the left. This is how I got started about, I don't know, 18, 19 years ago with this type of chip and this type of analysis. Amazingly and ironically, this company just got acquired 2 days ago, obviously, by Thermo Fisher, so you know, it's the end of an era. But I'm only showing you these gene chips to just show you an example of these high throughput measurements now, obviously, we have in biomedicine. But it's high throughput sequencing, it could be RNA-seq, it could be just DNA sequencing. Obviously, it's single cell work, it's proteomics, mass spec. We have all of these types of measurement tools now. Now, the one on the left, though, gene chips are so amazing, and they're so, I love them because they look like computer chips, and they're so cheap now that I can carry them in my suit. So I have one in every suit now, right? So I'm holding one in my hand here, right? This is a gene chip. And I only show this because data is often ethereal, right? You don't know how to look at data. Well, this is how you look at data, right? Big data comes from small packages, just like this. And now, these are so cheap. Obviously, we all use them in all of our institutions. But now, if you actually, if you're getting tired of measuring them one by one, you can measure them in the 96-well format. So that's this here, if I can get them collected here. So this is a 96-well format, okay? And that's so cheap I can carry that in my bag. And then when you get tired of measuring them in the 96-well format, you can measure them in the 384-well format, and that's in this hand. I cannot more clearly illustrate exponential growth in biomedical data than to show you these plates, right? When I got started 18 years ago, we were happy to have 60 of those single chips. And now people buy them like this, right? Now the company doesn't even exist, right? I cannot more convincingly show you how fast biomedical data is growing, just to show you these plates. Now, of course, the amazing part of these plates and these chips is it's not the chips, right? What happened in the early 2000s, 2001, when we got started in this field, is everyone started microarray experiments. And by microarray, I also mean RNA-seq and you know all those different types of technologies. But everyone started to use these. Everyone wrote papers about them. Here's a list of genes that are different in cancer. Here's a list of genes different in heart failure. And everyone submitted their papers. And what did the top-tier journals say? Let's start with the journals. And top-tier journals said, hold on a second. We're getting too many of these papers. No more papers with these gene chips. No more. Unless you share this data on the internet. You put this data out on the internet so that the reviewers can double check the math, kind of getting important to do that. We're getting closer to the clinic. And scientists can use that data for something else. And then we'll accept the papers, right? And it started around 2000, 2001, 2002. But the precedent for this goes back to GenBank, right? How many people use GenBank? Of course, everyone's used GenBank at some point in your career, right? GenBank, I think, is 33 years old now. Right? GenBank started with DNA sequences. First paper came out, ATCG. Nobody wants to type this in again. Right? So they're sending tapes to each other. This is way before 8-inch discs, 5-inch discs, 3-inch discs, CD-ROMs, DVDs. They were sending tapes to each other, right? Way before the World Wide Web. So the precedent is always there that if enough scientists use that type of measurement modality, you got to share your data with others. So fast forward to August of 2012. This article came out in Nature, featured my lab and others. In August of 2012, we hit 1 million of these chips publicly available. 1 million of them. From 0 to 1 million, exponential growth. Now actually, if you look really closely, looks kind of sad. It's slowing down here, right? And then you realize they only counted half the year, August of 2012, right? We're still doubling every 3 years. By the end of this year, we'll certainly hit 2 million, okay? So August of 2012 was like so long ago. I think we're at 1.8 million. I think we're over 1.8 million microarrays now publicly available, right? 1 million was so long ago. Still doubling every 2 to 3 years. And just to make it clear, here are the two repositories, Array Express, the Europeans, GEO run by NIH, NCBI. And you can go to either of these. If you just take care of the overlaps. There's about 1.8 million microarrays publicly available. Now I'm just showing microarrays, but think about dbGaP, thinking about all the rest of these repositories here. And all this data is sitting there, waiting for scientists to actually query this. So any scientist in our institutions, right? Let's say a cancer biologist or a medical doctor can go to these websites and they can type in, forget the scientists. Any high school kid, high school child, right, who needs to do a science project now, even a high school child can go to this website. Let's say she needs to do a science fair project on breast cancer. She can type in breast cancer, press search, and she can find and download almost 60,000 samples of breast cancer, about as easily as she could find a song on iTunes today. 59,000 samples of breast cancer. Now, what's so magical about breast cancer? What's so magical about 60,000 samples? If you haven't figured it out yet, 60,000 samples of breast cancer are more samples of breast cancer than any breast cancer researcher will ever have in their lab, right? Because every one of them has to contribute data into the repository. So whoever figures out the repository now has more data than any bench scientists. And yet I still run into informatics people just waiting for data, waiting for data, waiting for a collaborator to give them data. Here is all the data you can imagine sitting there. Now actually the number I like better than 60,000 is this number here, 2,400, 2,445. 2,400 independent labs have given data on breast cancer into this repository. Let me tell you what a ridiculous number that is. Let's pretend I'm a brand new researcher in breast cancer. I know nothing. And I'm writing an NIH grant. And you're on my review committee. And I propose to you, in my NIH grant, that I'm going to get 2,400 of the best breast cancer labs in the world to study their most characterized patients with breast cancer using the latest measurement technologies. And I propose to you that I'm going to get them all to give me that data for free. Okay? Some of you are already laughing. If I put that into an NIH grant, you would laugh me out of the room. Why would 2,400 researchers, the best researchers in the world, give you any data for free? Yet here it is, just sitting there, waiting for you. So when I think of public big data like this, the term I like to use is retroactive crowdsourcing, right? I'm retroactively crowdsourcing to the 2,400 labs. They don't even know that they're helping you. Actually, the 2,400 labs are helping you, and you don't even know they're helping you. And if for some reason 60,000 samples aren't enough for you, just wait another year or two. It'll be 100,000 samples. What are you waiting for? And this is breast cancer, colon cancer, prostate cancer, whatever disease you want, has probably already been studied by someone in the last 10 years. Now, if you have any doubts left that a high school child can do this, here's the high school child that did this, okay? This is a 17-year-old named Brittany Wenger, and about 4 years ago, she won the top Google Science Fair competition in the United States because she built an artificial brain, neural network, for diagnosing breast cancer. And she downloaded qPCR data, I think she downloaded some data from Geo, some immunohistopath data, and she built a classifier. The next year, I guess she was bored, so she was in the newspaper all over again for doing the same thing for leukemia. And of course, what I mean is if a high school child can do this, we all better be able to do this. But if we're professionals in this field, we better be way beyond this now, because this is what a high school child can do now. Simple classifiers like this are trivial, and the data's sitting there. Now it's a matter of taking this much, much further. But of course, even a high school child can do this. So in the remaining time, I'm just going to give you 3 stories of what's happened in the lab, 3 anecdotes, what we've been doing with this kind of data over the past 10 years or so. And each of these stories ends up with a kind of interesting outcome. You'll see as we go. So the first story really starts with my first big NIH grant. So my first big NIH grant was not just to go to these repositories for these microarrays, and don't just study cancer or diabetes, but go get every human disease that's ever been studied with these microarrays. Just go collect them all. And we do text mining to figure out what disease was studied. We human eyeball them to make sure we know which disease is which. And we started to collect experiments with disease and control, healthy controls. Disease and healthy, disease and healthy. And of course, I'm showing you these kind of colors instead of showing you the actual gene measurements, and we can compute these kinds of signatures. Marina did this work and made this slide. So we collect disease and healthy, disease and healthy. It's actually pretty easy to find a disease It's really hard to figure out if someone studied healthy samples, okay? What do I mean by that? We have 200 words for normal in biology, right? Normal, vehicle, wild type, control, time zero, margins, right? And then 200 other words. We wrote a paper on just the 200 words that people use. You can find our paper out there somewhere, just to show people we knew how to do this. So we can find like non-diabetes, non-diabetic is normal in certain experiments, so we collect all of these. Now, of course, like I showed you with breast cancer, it's not just the case that one disease has one experiment. For any one disease, you probably have dozens of experiments on, because plenty of people have been studying these diseases. So first thing we decided to do is, what are we going to do with this data? Let's make diagnostics. Diagnostics. What's a diagnostic? Helps the doctor tell, do you have a particular disease or not? Maybe it's a blood test or a radiology. Well, let's focus on blood tests here. And so here's just an example of the central dogma. It's really dangerous to show the central dogma so early in the morning, but here's the idea. Here, just to make sure everyone understands the concept of these kinds of chips, we have DNA, RNA, and protein. Of course, we all wish, look, I love protein diagnostics, but we know these chips are measuring RNA. Why do I love protein diagnostics? I don't mean mass spec. I'm talking about 1970s proteomics. That's what I love. There's tests called ELISAs, enzyme-linked immunosorbent assays. I love protein tests because they work in a desert, right? You don't need anything to get an ELISA to work. RNA tests, yeah, I guess they can work. Genomic Health is here. There are others. But you need a lot of setup to make sure the RNA doesn't degrade on you, right? If you just look at RNA the wrong way, it starts to degrade on you if you don't do proper things. So I know we measure RNA on these, but I love protein tests, right? I'm still amazed. I'm a pediatrician, very much part-time now. But when a kid comes with a sore throat and a fever, somehow you can stick a Q-tip in there, get enough antigen, it tells you right away that there's strep throat or not. I mean, that's a proteomic test, actually. It's really, really old, simple ELISA. But, you know, all we're stuck with are the RNA measures. Well, it's kind of simple and kind of stupid. If the RNA is changing in the disease, maybe the protein is, right? That's all the simple concept is. If we see the RNA changing, let's go chase down the protein, and not just the protein in the tissue. Maybe we can detect some of this protein in the serum or the plasma. All right, so we turn our crank. We have lots of data. We have lots of predictions. Here's a test. Maybe that's a test. That's a test. And then we came up with a prediction for acute myeloid leukemia, AML. Okay, not a good leukemia to have. They talk about putting a bone marrow into you when you get diagnosed with this, right? So not a good leukemia. So we said maybe we have a good computational prediction for a serum protein for leukemia. And what do we do? So let's go test this. Well, let's go to a cancer center at Stanford. I'm a member of the cancer center. Do we have a biorepository? Yes, we have a biorepository. Do we have any saved serum or plasma on patients with AML? And they said, actually, no, we don't have any saved serum. We saved the cancer cells, right? We saved the RNA, but they never saved the liquid around the cells because it takes more freezers to do that. Not everyone does that. That's okay, they said at Stanford. We are a major medical center. Go get the ethics board approval, the IRB approval. Go put up the posters. And you should have enough samples in a year to test your prediction. Now, what do we do instead as computational folks? We just Google for it today. And we find a store that will sell us the samples much faster. Here's a store, Conversant Bio. I see pictures of blood tubes. I'm in the right store. Browse by disease. What do you want to buy today? Bladder cancer, brain cancer, breast cancer. Okay, here's leukemia. What exactly do you want to buy? Do you want to buy viable platelet cells, T cells, negative selection from leukemia? Oh, here, peripheral blood, plasma. $55 each. How many samples do you want per patient? Add to shopping cart. I'm not making this up. This is for real. This is what we do. I'm not connected to this company except as a customer. We buy things like crazy from this company. What is this? These are stores that sell samples, right? So major hospitals in the United States, what happens is when you get a blood test, a clinical blood test, the lab runs the test. And then they hold on to the sample for about 48 hours because the doctor might add something else to the sample. Oh, I want this test too, I want that test. But more samples are coming into the lab, and at the end of 48 hours, what do they got to do? They got to throw out the sample, right? They got to throw it out because they got to make room in the refrigerator. And something magically legal happens on the way to the trash. It becomes a discarded sample, right? Now you can get consent or not, doesn't really matter. It's a discarded human sample. If you take off the old sticker, put on a new sticker, not put any identities on this sample. Me as a customer, I have to agree not to re-identify these people. I don't care who these people are. And this comes to you on dry ice in 72 hours. This is not the hard part anymore. Any of you computational people thinking that this is the hard part, I need to find a collaborator, I need to take something to the wet lab, this just comes in 48 to 72 hours and it's ready to go. Now, I don't want this to come to my office. In fact, in UCSF, I don't even have an office in. We have cubicles, barely cubicles. So I don't want this to come to me. Where do I get this shipped? I get it shipped to the core labs, the core facilities. Right? How many of you participate in core facilities, right? Informatics people are in core facilities. Nobody ever said that we can't use those core facilities either as computational folks. So we get the samples to come from one place, the antibodies to come from one place, other reagents to come from one place. They all come in the core facility, does the work for us. So we had this kind of model, right? So we're testing the proteins based on RNA measurements. We came up with a cancer marker that I'm not going to go into. It's a paint that you can apply in the operating room to glow. The idea is to make it glow to tell you where's the cancer, what's not the cancer for surgery. We came up with transplant rejection markers for a kidney transplant and lung transplant. In fact, it's a pan transplant marker for rejection. But the one I'm really proud of is this one, okay, on a disease called preeclampsia. Any of you heard of preeclampsia? Raise your hand if you've heard of this. Yeah, only a handful of people. Preeclampsia is when the blood pressure goes really high in a pregnant woman. Some people know someone who's had this. Maybe you've had it yourself. This is still how a lot of husbands lose wives around the world. I mean, this is actually a severe problem. In the United States, the only people who know about this is if they watch this show called Downton Abbey. One of the characters dies of preeclampsia. Sorry, spoiler alert if you haven't watched it. But this is the only reason Americans even know about this disease, right? But it's a very annoying disease because it causes a lot of this mortality, morbidity. The obstetricians, the people who deliver the babies, they have it easy. They can just say, take the baby out, the mother gets better quickly. But we pediatricians then have to keep these tiny babies alive for months. And there's no diagnostic for this. Forget about a prognostic, there's not even a diagnostic. The test we're using is 100 years old, looking for protein in the urine. And so we can do better. So Linda Liu started this project looking for public data, RNA data, to see if we can do this. And if you just search for preeclampsia or preterm birth, 266 experiments. Here's 300 samples, 250 samples, 190 samples, all human. How many more micro experiments do you need to get started here? Now, I'm not going to show you any methods in this talk, right? But how do we do this? Just imagine a big Venn diagram. You know Venn diagrams, right? And I'm just taking the best ones of these experiments and figuring out what's in common, right? I don't trust any one of these experiments, but I trust what I see in common, right? If the top 5 or 6 or 7 all show these same RNA markers are going up, then maybe we can chase down the protein here. That's the idea here. And so we do this, and we find, for example, a marker like this. Hemopexin is up in the serum of preeclamptic mothers compared to the normals here. And by the way, we bought those samples, the first pass, we bought those samples off the internet, $45 each. It's amazing how much easier it is to find collaborators after you've done some wet biology, right? If even the most preliminary experiment works and then you go to your local people, believe me, they want to work with you because they know that you're the one that's going to run with the story. You're not the middle author and that kind of thing anymore. It's your story. You already got some data. Everyone wants to start to help you now. Now, this one's a really interesting story because Bruce Ling did the proteomics work, Linda Liu did the computation work, and Matt Cooper got involved because we started a company on this one. So you're going to see 3 arrows like this in my talk here. We always start with the unmet need, right? You got to know what's the question you're trying to ask. That's the hardest part. We need a diagnostic. Public data is available. We had March of Dimes Center money, seed money to get started to design the diagnostic, analyze the data. We got a Spark grant, which is like an internal seed grant at Stanford, $50,000 to actually test it. And we spun out a company, Life Science Angels and others, put more than $2 million to start this company. Now let me be really crystal clear for a second. I'm not showing you $2 million to brag about it. I'm showing you $2 million because I fundamentally believe this is the new way science will continue out of academia. You all know how hard it is to get a brand new $2 million NIH grant. But once the paper is written, and if there's value for patients or doctors or families, spin it out of the university and get private dollars to fund this. The science continues in the company now. We don't know if this is going to work or not, right? You're going to do experiments there. But now it's being funded on private dollars, and your lab moves on to something different. And that's the idea here with raising money and starting a company. The science continues that way. Now this one, as Ben was alluding to, is unbelievable to me because we launched a multi-center trial and we already got acquired. The science hasn't even finished yet. The company wanted to buy us. So we sold the company to a company called Progenity down in San Diego and already exited. Inventors happy, investors happy. Of course we can do more of these. And I'm giving away our secrets here. I'm telling you how we do this. Because there's so much unmet need in medicine, there's such a wide space for diagnostics here. That's how important. So we went from public data to an acquired company in 24 months. Okay? Total company size, 1 person. Everything else was outsourced. 1 person company, Matt Cooper. And that's it. Now he's joined the company there. So that's one example of diagnostics. Let's start the second story. Sequencing. Obviously, sequencing revolution. You know this. We did this genome project in factories like this. This is the Whitehead Institute, where you have robots picking clones, running gels. And of course, we're heading towards sequencers the size of a USB stick. Right? I think the MiniION first data is publicly available now, I think at EMBL. It's a no-brainer that this is going to impact medicine. It's a no-brainer. Don't keep asking, is it going to impact medicine? I'm not even going to say in my lifetime. By the end of this decade, sequencing is going to revolutionize medicine. It's a no-brainer in the next 5 years. Don't even keep asking, is it or is it not? In fact, so people say, what's the current cost? Right? The Genome Project took 13 years, cost $3 billion. I think now the best price is about $1,500 for a genome. And the speed record now I think is 27 hours. You can go from actual heel stick of a brand new baby born to some gene annotations from exome sequencing in about 27 hours. Right? It's amazing how fast it's going. And still people think, well, the genome's going to be $100,000 or $10,000. My favorite is this one. Complete Genomics, this was back when they existed. Cliff Reed had this our blog post back in 2011. He was so annoyed because people kept saying, well, it might cost $10,000 by the end of the decade. And he said by 2020, he was predicting it's going to come down to $33 per genome by the end of this decade, right? It will cost you more than $33 to have dinner around here, let me tell you, okay? That's how cheap this is going to be, right? $33 is nothing, right? You know that. $33 per genome. And what happens the day after it becomes $33 per genome? The following year it becomes $0, right? And then the following year, it'll be $- for your genome, right? So some of you are laughing. How do you get a $- for your genome, right? I'll just give you one analogy. It's actually in Wall Street Journal today. Good example. There are car insurance companies, okay, Progressive in the United States, a couple car insurance companies in the United States. They're crazy people, these car insurance people. They have ads that are just ridiculous. But these car insurance companies in the United States, that if you agree, they will send you a gadget that you put under your dashboard, and it tracks how you drive, okay? Now, some of them have GPS, some of them don't, but they see where you're driving, and it see, do you accelerate or do you brake? And if you agree to have that gadget, and you don't drive like a maniac, they will reduce your car insurance rates. Can we imagine any health insurance company doing this by the end of this decade? If you agree to get a genome and you agree to compensate for any risk factors, they'll reduce your health insurance rates. Of course, we can imagine health insurers doing this. I can imagine them all doing this in 5 years as we get into a new mode for paying for healthcare. So that's a $- genome that people will pay you to get this if you agree to compensate for behavioral change and things like that. That's where I see things going. We were lucky at Stanford because we got to analyze the first patient to show up with a genome, a whole genome. Now this is a very unusual patient, Steve Quake. He starts sequencing companies on the side, but he showed up because here he is in the family tree and his nephew died of sudden death. Age 19, did not wake up one morning. So he became the first patient to show up with a genome. Steve Quake went to his cardiologist, Ewan Ashley, and said, "Doctor, am I going to die of sudden death like my nephew? And doctor, here's my entire genome," okay? And so this became an unbelievable story. It was in the Lancet back in 2010. I think it was one of the most highly cited papers in all of medicine that year. We were lucky to be a part of this. And what did we figure out for Steve Quake? Well, Steve does not have any known variants in known risk factors for sudden death. He has a variant in a gene that might give him a heart attack later in his life, but he doesn't have any known variants. So the heart was okay. But now that we have his genome, what else can you do in medicine with a genome? Well, Russ Altman could give an hour-long talk on pharmacogenomics. All the different drugs, 150 drugs now that we can figure out the dose. Is the drug actually going to work? What the side effects might be. 150 drugs, including clotting drugs, statins, and all the rest. This could be an hour-long talk, just this. I'll just show you one slide. And our lab was stuck with everything else in medicine. Okay, heart, done. Drugs, done. Everything else in medicine was left to us. And so how do we accomplish this, right? So back in 2008 or so, 2008 is when we really started. I knew someday a patient was going to show up with their genome to our medical center. And so I started with a high school child from San Jose, high school kid. He came to our lab. We gave him 50 papers. And I realized there's no master list of every SNP and every disease. There's no master list. SNP, disease, SNP, disease. There are a couple interesting ones like the GWAS catalog, but they have problems I can talk about. So we gave 50 papers to this high school child, and we said, I want you to curate these 50 papers. Write down every SNP, every fact that you get from the 50 papers. So we figured out a data model, and we figured out he could do 50 papers in a summer. How much do we need to just read the entire literature of genetics? And that's what we did. We reread the entire literature of human genetics. So what do I mean by reading? Here's a paper, Association of IL-23R. This is published in 2008, Journal Inflammatory Bowel Disease. You can guess what kind of diseases they look at. Here are some diseases. 700 patients, 9 loci. And we write down whether it was a GWAS, what kind of study design, disease phenotype, population, the gender breakdown, all the alleles, the p-values, confidence intervals, odds ratio. We write down every single number from a paper like this. And we map it to a standardized vocabulary called UMLS, which is a just wonderful product from the NIH. Here's an example of the tables. Here's RS1004819. This is SNP ID. Humans have a C or a T allele. We write down all of these numbers, all of these frequencies here. I love natural language processing, but NLP is terrible like this on tables. You know that. I know that. You can't even figure out where the tabs go, right? Forget about like which table, which column has what. So you cannot NLP this, right? It's just impossible. You've got to manually read every one of these papers. Here's a paper came out the following year in 2009, Journal of Rheumatology. You can imagine what diseases they look at there. Here's a disease, 8 SNPs, 2,000 patients. This table is sideways. You have to rotate it. Here's a SNP, R1004819. Here's an A allele. And we write down all of these facts from this type of table here. We just back compute how many patients had which frequency. Now, if you were paying attention, and you could see the slides, you'll see I showed you the same SNP in 2 papers. All right, here's RS1004819. This paper says humans have a C and a T. And this paper says they have an A. They can't both be right. What are the alleles for 1004819? Well, you got to look each one up in DB SNP. And you realize humans have a C and T, and that Wellcome Trust paper, that was actually wrong. And then you realize 11% of all papers in human genetics make this error. This is not a strand problem. They're reporting on the probe they use, which is a reverse complement to one of the strands. It's a complete error in these. So you got to fix all of these. And then you realize NLP can never do this. So we've done that, right? And all these papers, all these findings are locked up in the publications. So here's one paper, here's 6 papers, here's 100 papers, here's 1,000 papers. So I have a script that just generates these from the PDFs. And now I think we've blown past 20,000 papers, more than almost 2 million records, 500,000 SNPs, and we got 7,000 diseases. It took a couple years to do this, but it's not impossible. Even we as academics can do this. And how are we going to do it? We outsource the whole thing to a company in India. Got like 100 geneticists and bioinformaticists just reading papers and curating. They have a whole data model. Company's called Optra, and that's how we did it, right? Because I don't want to have curators here. I want to go with the database and figure out what we can do with this, right? I don't care about hiring the curators, but find a company to do that. Here's an example of what we call a riskogram. This is from the Lancet. Here are all these different diseases. And the black arrows indicate what we thought the risk was based on epidemiology, and the red and the blue indicates a new post-test probability given the genetic risk. And we have all sorts of fancy ways to show that now. What can you actually do about the disease given all the different behavioral changes that you can do on the edge here? Let's skip forward a little bit. Now, if you really want to get into sequencing, if you really don't know how much sequencing can change lives here, this is the kid to look up, Nicholas Volker. How many of you have heard of this story? Yeah, very, very few. This is a Pulitzer Prize winning story. Look up this story, okay? First child saved by DNA sequencing. When Francis Collins, the director of NIH, goes to Congress, he talks about this child. So this child, back in 2011, was 5 years old, and he spent the entire first 5 years of his life in the hospital because he kept killing. Parts of his bowel were dying. The cells were just dying. He had surgery after surgery, just taking out pieces of his bowel. He was only 5 years old. This child was going to die. They had nothing they could offer, except they said, let's just do a DNA sequencing on you and the parents. And they found a novel variant in a gene called XIAP1. It's a random gene, but that gene is actually expressed in lymphoid tissue. I said, maybe there's something going wrong with the immune system here. They did a stem cell transplant in this child, and now he's out of the hospital. He goes to school. He actually eats regular food. Now, he's not out of the woods yet. He still has a lot of problems, but this is the first child saved by DNA sequencing. It used to be that when we wanted to come up with a new name for a Mendelian disorder, you needed 100 children, then we got it down to 10 children, and now we can get it down to N of 1. Here's a child with a disease no one's described before, but we have his DNA and the parents, and we can figure this out now, right? And now all of our children's hospitals get a lot of these children, actually, a couple dozen for all the good ones. Each one gets a couple dozen of these parents and children trying to end this diagnostic odyssey. Is it this? Is it that? Just get to the answer and do sequencing. This is revolutionizing. It's a completely amazing use for whole genome sequencing in this population. The other one I'm going to talk about is obviously cancer genetic sequencing and neurological disorders. We have lots of different companies now offering cancer sequencing and neurological sequencing. This is actually clinically approved now and can be ordered. I'm conflicted with this one, but not the one in the back, so we got lots of different companies here. And what do you do in Silicon Valley when you have that database? You start a company. And so this is 4 faculty founders, me, Russ Altman, Ewan Ashley, Mike Snyder, and then our CEO, John West, who used to run Celexa, now he's running Personalis. We had the unmet need to analyze medical genomes. We curated all this work. We tried twice to get NIH funding to build our database, failed. Just even with the 2 percentile score, we couldn't get money. So we built it, we got our own money, we built a database, we launched a company, and today I think it's like $75 million in from investors. I know 150 people in the company down in Menlo Park. You know how hard it is to get $75 million in an NIH grant? The science continues in the company, right? Do you see the theme here? This is how the science is going to continue out of academia. This is how you're going to have impact and scale. Not just to keep doing the simple thing again and again in your lab. Get your lab to work on the next big thing. That's what we're supposed to be doing in academia. The final story is going to be on drugs. Obviously, we have this big JP Morgan meeting going on in San Francisco. There's diagnostic companies, but a lot of them are the drug companies. And the drug story is a mess. It's a total mess. I know FDA is approving more drugs than they used to, but it's still a mess. If I ask any of you, in fact, if I ask lay Americans, how much does it cost to develop a drug? It's amazing. Most of lay America knows nothing in common, but they know this number. It's $1 billion to develop a drug. How do they know this, right? I mean, they don't know anything else in common, but they know this $1 billion in 10 years because it's advertised on the Super Bowl. The pharma industry puts these advertisements. $1 billion. That's how you justify the prices, right? $1 billion per drug is an underestimate. That's the wrong number, right? How do you get the right number? Well, it's simple math, right? Very simple ratio. Total pharma R&D spending divide by the number of drugs, right? How much did you spend? Divide by the number of drugs. And if you do that math, it costs between $3 and $12 billion per drug. Not sustainable, okay? This is not sustainable. There's all sorts of weird pathologies in these lists here, right? With Pfizer, AstraZeneca. Pfizer in the middle here, it's $7 billion. Now, I'm not a strategic consultant or anything, right? Pfizer in the middle at $7 billion was trying to buy this company, AstraZeneca, at $12 billion, right? If I were spending $7 billion per drug, I'd try to buy one cheaper than me. Why would you buy the most expensive one? I don't know. Has to do with pipelines and all this, right? But that's how crazy this list is. Not going to be sustainable anymore. So while we're building out the database for the diseases, we started to see people depositing drug data into the public domain. Most notably, this is Justin Lamb and Todd Golub at the Broad Institute with the connectivity map, and now the Links project. Lots of data on drugs. Here are cell lines with drug, without drug, with drug, without drug. So on the left, we have disease, no disease. On the right, we have drug, no drug. And they're completely different tissues, right? Completely different diseases. But we start to see the signatures, and we start to see if we can put these two together. And to make the informatics very simple, I'm going to use my arms. If I have a disease where this gene goes up and this gene goes down, and I can find a drug that can make this one go down and this one go up, maybe there's a match there, right? That's with 2 genes. Imagine 20,000 genes. Imagine Pearson correlation, Spearman's, Kolmogorov-Smirnov's. There's a lot of ways to do this, right? I'm not saying we have the best way. We have one way. There are lots of ways to do drug repositioning. Now here, you can use any drug, but we had a lot of fun going after the drugs that were already used for something else, right? Here's a drug, it's used for that, but we think it could be used for this, right? That's the idea here. So we got lots of ideas. We turn our crank, we got lots of ideas. And we said, boy, let's just go run a clinical trial. But actually, before you put these predictions into humans, you got to run a mouse model. And all my mouse friends at Stanford, they start to scatter, right? I mean, they know we're going to make a lot of work for them, right? A couple of them stuck with us, Alejandro Cicardero and Jay Peshvika and Julian Sage. They helped us a lot with this kind of testing of these drug models. But now, if we really, really need to have a mouse model run for us with a drug, we go to assaydepot.com, right? This is the store. What do you want to buy today? Biology, chemistry, DMPK stands for drug metabolism pharmacokinetics. If you don't know what that stands for, don't click there. Pharmacology, toxicology. Let's click on pharmacology. What type of mouse do you want to run today? Bone, cardiovascular, dermatology, diabetes, urinary, infectious disease, inflammation, neurology, cancer, eye, ear, pain, respiratory. What type of mouse do you want to run? I'm a diabetes person most of the time. Let's click on diabetes. Here's the OB diabetes model. I'd like to purchase 16 mice. Maybe I want 2 groups of 8 or 4 groups of 4. You send them the drug, it's blinded. If you read here, it says this, what is this OB? If you read, it says this mouse has been it's been eating a lot and getting diabetes since 1951. This is a standard model of American diabetes, right? You eat a lot, you get diabetes, right? It's not the best model, it's not the worst, it's one everyone uses. And in 28 days, they'll give you the fasting blood sugar, insulin tolerance test, glucose tolerance, whatever you want to see if the drug is working. Now, what am I covering up here? The price, $9,000 for this service, 9-week turnaround time, add to shopping cart. Okay? I'm not making this up. You can buy an entire mouse model off the internet with a credit card today. Now we do this like crazy now in my lab. We just do this like crazy. Of course, we don't always have to use a company. We can use our friends, collaborators, core facilities, but this is how we get the experiments done. Now some of you are really wondering, how do you trust the quality of this work, right? You're outsourcing this, how do you trust the quality of this work? Actually, when you hit add to shopping cart, you see there are 133 companies or labs willing to do this experiment for you. 133 of them. Well, if you need them to be USDA approved, click here. There's 4 of them. ISO 9001, 7, AAA 28. So you click on the certifications, you only see the companies that actually have that certification. I wish it were that easy to find the right collaborator on my academic campus this way, right? So I knew someone that actually knew how to do the experiment was doing the experiment, so some random undergraduate, right? But here I can click on certifications. You also note that outsourcing doesn't always mean offshoring, right? Because you have Maryland, you have Wisconsin, so these companies are all over the world. You can pick and choose where you want to send the business. Now, some of you are still wondering though, right? How do you trust these companies? All that computer stuff is kind of interesting, but this seems like the most important part. Because based on this, you're putting the drug next into humans. So how do you trust this? I'll simply say, if we really, really need to know if this drug is working in the mouse model, just order 2 of them from 2 companies. Hell, at $9,000 each. Order 10 of them from 10 companies to do this experiment. Now, why am I saying it in this kind of weird way, right? How many mouse researchers do you know, when they get a mouse experiment to work, do they go across the hall and get a completely independent team to repeat the experiment? You know nobody does that. Yet I can get as many independent teams as I want to repeat my experiment. I just need more money. So what I've done now is I've changed the quality problem to make it a function of dollars. You have more dollars, you get more companies to do this. Right? So the quality problem completely evaporates. I don't trust any one of these microarray experiments, but I trust what I get in common. I don't trust any one of these companies, but I trust what I get in common. And because we have so many validation methods now, this is how we do it. So we typically order one from the East Coast and one from the West Coast. This is an example of one that we did. This is one that we're most famous for, is a seizure drug, an epilepsy drug we predicted works on inflammatory bowel disease, Crohn's disease. And these are my famous rat colonoscopies, okay? These are actually alive rats where you can put a fiber optic and see how the rat is doing. So this is a normal rat colon here. This is a rat that's given a chemical to have this disease, inflammatory bowel disease. And this rat is all better after this chemical and after the epilepsy drug here. And this example, this is a experimental model actually that nobody on our campus knew how to do. So I'm not just outsourcing to save money, I'm outsourcing to find the best damn data in the world. There's a company that knew how to do this. None of our collaborators knew how to do it. But the one I'm most proud of is this one. Okay, so this one came out about a year ago. This is an antidepressant, okay, a drug for depression called imipramine. We predict it works on small cell lung cancer. So humans smoke to get small cell lung cancer. This mouse, you knock out these 3 genes, it gets lung cancer. See the nodules here? And this mouse is all better with imipramine. Now, we don't use imipramine anymore in the clinic. It's in the pharmacy. We don't use it anymore. We don't use it because it has side effects. We use serotonin reuptake inhibitors now. Imipramine has side effects. It has 2 major side effects. It can make you really sleepy, and in some people, it might give you an arrhythmia, set you up, called QT prolongation. Actually, neither of those 2 side effects sound as bad as lung cancer, 5% survival rate in 5 years. And the cancer is melted away, it's gone. No more cancer left in this mouse. Now, why do I show this one? We've done a dozen of these. Why do I show this one? I love this one. Because we went from public data, we had the prediction, we test cell lines in a petri dish, I'm not going to show you that. We get this mouse model to work, amazing pictures. 15 months after the computer prediction, we got ethics, IRB approval. We launched a clinical trial on this one. Humans are being dosed on this now. 15 months. Total cost of the trial, $50,000. Not even $1 million, not even $100,000. $50,000 to launch this trial. Now, I don't know if the drug is going to work or not, right? This one might fail, but we got to do more of these. Because we cannot wait for the pharma and biotech industry to test all of these cancers. There are not enough people there either. It's going to come down to us to do this. And it's in our capacity to do this, to get the predictions and to launch this. And what do you do in Silicon Valley? You got to start a company. And this is the third one. I'll just spend a moment on it. The unmet need is there. We had NIH grant money to build the databases, start the company, and now more than $5 million raised. And I'm really proud of this company. They announced a couple weeks ago that they have a partnership. They're developing new drugs for a company called Allergan. So Pfizer Allergan, if they merge, will be the largest pharmaceutical company in the world. Yet this is a garage biotech, right? Just a couple people in a conference room developing new drugs for a huge company like Allergan. They just announced another deal this morning. All right, in the last few minutes, let's get out of the molecular realm for a second. How many of you have these types of devices, Fitbits, watches? Anyone in this audience with a Fitbit, a wearable? Boy, just 2 people. Wow. The standard for Silicon Valley is about 25 to 33%. So you're underrepresented actually right now, okay? So this is a field of quantified self. Now I used to laugh at these people, right? They want to count everything about their body, what comes in, what comes out. You know, it's kind of weird kind of group of people. I used to laugh at these folks until December of 2012. December 2012. I came back from PSB, you know PSB, the bioinformatics meeting, right, in Hawaii. Come back from Hawaii. I step on a scale and I weigh 247 pounds, okay? And I realized when I hit 250 pounds, they can measure me in tons, right? That's a quarter of a metric ton. So what the hell happened to me, right? How did I get like this, right? So I bought everything Fitbit makes. Personal big data, lost 50 pounds, right? 50 pounds in 2 years. Because I write down everything I eat, and more important is the scale. That scale is amazing. Can't cheat the scale. Every morning I have my weight. I carry it in my carry-on bag and everything. So I'm a big believer in this type of phenotyping and behavioral change. Okay, I love this kind of thing. But here's the important part. Every one of my friends on Facebook knows I lost 50 pounds. But my doctor, who it's been written in the newspaper, my doctor, Sutter Health, spent $1 billion on their electronic health record system. $1 billion. Is the last to hear that I lost 50 pounds, this major health intervention, because their $1 billion system doesn't talk to my $40 gadget. Can you smell the opportunity here, right? Can you realize How much is going to change in the next 5 years? That if the doctors don't get this, we as patients are going to cut them out of the loop. And they're not going to get involved in our care anymore. So you can see there's dozens of startup companies just in this space now trying to figure out how to get this data. So much data being collected, not in this crowd, strangely. So you're behind. You don't have these types of measurements. Let's skip ahead a little bit here. Let's find it. Okay, I'll spend a second on this one. What's the next big open data? Half my lab basically works on clinical trials data. Okay? So you know the famous saying to skate where the hockey puck is going? This is where I see the hockey puck going. What's a clinical trial? A clinical trial is the most expensive experiment in the world. Right? It's why it's costing us billions of dollars to develop drugs. Most expensive experiment in the world, half of clinical trials fail. And when we have this weird culture that when a trial fails, we don't even write a paper about it, right? Forget about releasing the data. It's going to change. It's going to change. The European FDA, called EMEA, is requiring raw clinical trials data release. FDA is going to get there as well in the United States. What can we do with clinical trials data? Well, we can start to figure out, did this trial actually fail? Maybe it succeeded. Maybe we can do patient subsetting. But here, we run this repository for the Allergy and Infectious Disease Institute of NIH. Go get an account and download 100 raw clinical trial data sets. Learn how to study clinical trials data. I see a lot of value, both commercial and academic, coming out of this. So we give out 100 trials from this repository here. And so much more that I don't have time to talk about. Microbiome, electronic health record data, and all the rest. A couple last points. I'm pleased that we're in California. You're all visiting California. California, as usual, is ahead of the rest of the country. We launched about a year and a half ago a new initiative for precision medicine. I'm pleased that I get to work with the governor and the state capital on this, where the state itself is now investing in precision medicine. Just like they did in stem cells 15 years ago, now we're investing in precision medicine. It's going to be the next big growth opportunity for business and for academia, so we're doing a lot here. Why am I so happy to move to UCSF? I get to build out this brand new building. We're actually temporarily in this brand new building until they build our brand new building. Amazing, it's right between Illumina and our brand new hospital. You couldn't want a better location for a computational biology group in Mission Bay, up the road in San Francisco. But we also get to run the repository for all the patients seen in all 5 University of California medical centers, UCLA, San Francisco, San Diego, Irvine, and Davis. About 14 million patients get their care in the University of California. So it's 4% of the US population. And we're building out the data warehouse for every byte of clinical data on these folks. So that's a new challenge for us. What are we going to do with that data? Data is still going to be there. So our goal, really, as I recruit a dozen faculty over the next few years, is to build the strongest team in the world in medical data analytics. And finally, let's just close it out here. We're so used to, you're visiting Silicon Valley, most of you, we're so used to these kids around us here in Silicon Valley, and I'll admit they're annoying kids. They start these amazingly high-value companies in their garages, right? Apple and HP started in a garage. They're famous garages over there. Or they start these amazing companies in their dorm rooms, right? Like Yahoo, Facebook, Google started in dorm rooms, right? I sincerely believe the next Genentech, the next Amgen, the next Pfizer is going to start in a garage. You don't need $100 million to start a biotech company. When you go home from this meeting, you already have nearly 2 million microarrays at your disposal and all these mouse models that you can buy with a credit card. Now, maybe you can't afford $9,000 in your credit card. I understand. That's why you have angel investors, seed investors. What else do you need to get started with a biotech company? And this, I think, is the big deal. The next big thing is going to be these garage biotechs. Just like you need you can get an app done at a desk. You can create these kind of companies. I think we can do this for biotechs too. So in the end, what is big data in biomedicine? Is it about algorithms, programmers, databases, high-performance computers, Hadoop, mobile? Yeah, it's kind of about all of these things, but big data in biomedicine is about predicting diseases before they strike, explaining rare diseases that defy experts, finding drugs for diseases that lack attention because there's not enough pharma and biotech companies out there, making sure we do the right safe thing for patients, and it's an amazing platform for biomedical innovation and entrepreneurship. I can sum all that up with one word: hope. We are the hope for the future for patients and families. I am not yet convinced the pharma and biotech industry is going to figure all this out. I consult for them all. You saw that on the second slide. It's going to come down to you and me and us to figure this out for our patients and families. And I encourage you to think about the translational value of what you're doing. Can't do any of this work without a long list of collaborators that I just don't have time to even mention here. And can't do any of this without generous endowment support now from the University of California, San Francisco. My lab has 17 NIH grants from these 9 institutes of NIH. These 4 give me more, these 5 give me less, but I still love them. Marcia Dimes, JDRF, HP, HMI, a lot of disease-specific foundations that give us money to work on their diseases. I always thank my admin and tech staff. I would never get a grant or paper out the door without them. And I always thank the folks that mentored me and recruited me. And I always thank my wife, a molecular biologist and biochemist. She reads every major paper and grant that comes out of my lab. She starts companies with me now. And she lets me go all over the place to give talks like this. Thank you very much.
Thanks, Atul, for the excellent talk. We do have minutes for questions.
Sure, there's a question in the front here.
So how do you...
They're recording it, so you're going to want a microphone.
So can you comment out the privacy concern about sharing data as a consumer
Yeah.
to this kind of platform and what's the impact with the open data?
Yeah, so the privacy concern about sharing data and impact on open data. So a lot of laws are changing now. The simple thing to say is the common rule, which enables us to use samples, is changing, it's getting rewritten. So we're entering a mode now that we're going to have to really pre-consent patients for a lot of research reuse. Now, at our institution, we kind of already do this. When you sign in to any major academic medical center, you're already signing consent in a big blur of text is that we can reuse your samples, reuse your data. But things are going to get a little bit more strict now that we're going to have to actually tell you exactly what study we're going to be using your samples for. So law might change here. The data, so a lot of this is available, but also under lock and key, right? So for example, the genetics data that we can use to re-identify someone, presumably, you need to go to dbGaP. It's not as easy as Geo to download data. Any high school kid can get data from Geo, but dbGaP, you have to be a vetted investigator. Some of them you had to get IRB approval. You had to get approved by the research committees. So some of them are appropriately harder to get to, but you can still get to it, but you got to follow the rules. So in some ways we're protecting, we want to make sure that people are protected in that way, but the laws are constantly going to be in flux now. Other questions? There's a question on the table back there.
Bigger general questions. How do you define the difference between researcher and entrepreneur?
How do you define the difference between a researcher and entrepreneur? Okay. Absolutely, it's a great question, great question. To me, I start to realize, my wife taught me this about 11 years ago, okay? She taught me something even before we got married, right? She's really smart. And she taught me there's no difference today between a good grant and a good business plan, okay? Now think about when you start a lab, right? When you start a lab, they're giving you, let's say, money, academic lab, they're giving you money, let's say, to keep you going for about 3 or 4 years. And if you don't change the world in 3 or 4 years, you're not going to get any more money from your academic institution. That sounds kind of like a startup company, right? That if you get a pot of money, get started, if you don't change the world, you're not going to get more money. You're not going to raise your Series B. In fact, what we realize, so we in academia now, if you think about what my job in academia now is, I'm a serial idea person, okay? What does that mean? My job is to keep coming up with ideas. Some of my ideas are so long range that I'm going to write an NIH grant. And maybe I'll get money, maybe the experiment will work, okay? Some ideas are so immediately going to help patients that I'm going to start a company on that. And our academic institutions let us do that. Basically, every academic medical center in the United States lets you spend a day a week starting companies, okay? There are all sorts of different vagaries in the rules and the laws, but essentially every university lets you spend a day a week doing it. Most faculty don't do this. But today's exercise now is how do you convince a room full of people to give you money to execute on your ideas? What difference does it make if the room is in Bethesda at NIH or Sand Hill Road with the VC community here, right? It's the same exercise of how do you convince people to give you that money. So I think there's a lot more in common here than most people actually think. The question in the back, go ahead.
One question is, when you mentioned that child was cured like a rare disease,
Yeah.
could you give some insight why that was possible?
Yeah, so exactly. So why was that child cured? Now, just to be really clear, this is a lucky kid because a large number of these kids, we actually get the variant and we don't know anything that we can do, right? We might get some more clues. This one, in some ways, got lucky. Maybe they were thinking it was already an autoimmune process. But I guess seeing this variant in that gene really convinced them that there's something wrong with this kid's immune system, and that really suggested them that they should do a stem cell transplant. So that's what they ended up doing. I want to be really clear though, the vast majority of these kids, we don't have that kind of insight. That takes time to figure that out. So we need to do models, we have to introduce site-directed mutagenesis and cell models to try to figure out what the physiology of the story might be. But this is an amazing kid that really got lucky in that way. And I also want to make that kind of point here. In precision medicine, right, I didn't even use that phrase really, precision medicine, we have all these diagnostics. The rate-limiting step is going to be the therapeutic side, okay? That we are going to get a lot of diseases and all sorts of subtypes and sub-subtypes. What we're stuck with right now is we don't have enough things we can do to patients, right? And a big chunk of those are therapeutics, whether it's biologic or small molecule or even behavioral change. We're lacking on the intervention side. So if you had to pick between diagnostic or interventional, I think there's a lot of value going in the interventional side now.
So one, you do many labs, right? So you bypass somehow the normalization, standardization of data. So you want comparability across different labs. Is that the right way to see it?
Say it again, I didn't get the question.
Because when you get data from different labs,
Yeah.
you don't know how they produce them.
That's right.
And then, but you bypass this standardization.
That's right.
What are the issues through this different?
That's exactly right. Now, yeah, whoop, I think that, okay, so that's exactly a great point. I actually don't need them to all run the same experiment. In fact, I prefer that they don't, okay? So in general, for example, they have breast cancer and normal, breast cancer and normal. We did this with diabetes. Diabetes, type 2 diabetes and normal, type 2 diabetes. But I don't care if you ran rat and mouse and human and liver and fat and beta cells. I actually prefer them to be different experiments, and then if I see something in common, I know this is a more robust finding. So this kind of thinking came from a Nature Methods paper. I can send you the reference if you're curious. I don't have time to show it here. But this is where I got this kind of inspiration, that I'd rather have 100 different ways to do the experiment and figure out what's in common than to have 100 labs do exactly the same experiment, get the same answer, which actually doesn't generalize. So there's some thinking there.
All right, we have to
Thank you.
wrap this section. Thanks, Atul, again.
Thank you very much.